Hey, Dr. Stansel, it's Ashley Allen. Week 3, lab number 2. This is Bacterial Morphology and Staining Techniques. There's actually a lot to say in terms of bacteria. So when I say morphology, let me explain what that means. Morphology is the shape, size, and arrangement that you find bacteria in. In one sense of my mind, I think that texture must come into play, but I will leave that aside because we haven't gotten there yet, I think, in our reading. So shape, size, and arrangement. So there's three categories. There's cocci, which are round or spherical bacteria. They're the smallest of all three. And when they replicate, they can clump together like grapes. So I'm actually going to show you a picture, which is in our lab. So similar to that, they clump together, fairly small. And then the second one is bacillium, which these are like longer rod-shaped bacteria, and they don't necessarily clump together, but they link together end to end, so it looks like a chain. So I'll show you that one as well. So here's bacillium. Okay, so then the third one is spirillum, which is like spiral shape. And the difference with these guys is they hang solo. So you can see like a lot of them are not together here. So those are the three shapes. But then you think, well, how do we know they're three shapes? Well, that's where staining comes in. So there's different types of bacterial staining techniques. We use a few of them in our labs. In particular, in this lab, we use direct and negative. But what we did, so a simple stain is just like one dye, and it can stain either the cell or the background. Very simple. A direct stain is only stains the cell, so the background is colorless, so you just see the little cells. A negative stain is the cell itself is colorless, but the background is darkened. And then the differential stain are two different dyes that are used to see the cell, but also any other structures attached to the cell, like flagella. So those are the four stains. So when we get down to stains and bacteria, we've all talked about from week 1 gram-negative and gram-positive bacteria. So we use these because there's two different types of bacteria, but we don't know which ones, these two different types, we don't know which one's which until we actually stain them, right? So a gram-positive has a thicker wall. It has that peptoglycan wall, and the gram-negative has it as well, but in the gram-positive, it's fairly thick. And so what happens, the first thing we do, which by the way, I just took a swab of my cheek that I'm going to show you here. But the first thing we do is, you take the swab, I let it dry. This was of my cheek, and I'm going to fix it with heat. Make sure it doesn't leave the slide. We don't want it off the slide with all the stains that we're going to use, and we use a little water. So this is the first process. So I'm going to fix it there, get some heat going, make sure the bacteria is fixed onto my slide there. And then we start with our dyes. So the first one to use is the crystal violet, right? So crystal violet sits on there for about 1 minute. So we'll put a few drops on there. And while we're waiting, let me tell you what happens. So gram-positive stays crystal violet, and gram-negative does not. So we do the crystal violet, then and everything turns violet. Then we do the iodine, and that kind of locks the violet in there. Then we do the decolorization process. Step 3. What happens there is it takes away the violet from the gram-negative because it doesn't have that extra thick wall that the gram-positive has and that holds on to it. So then when we add the saffron, which is step 4, this dark red color, the gram-negative just soaks it up and it becomes pink. And although the gram-positive has the saffron too, the violet color is just so much darker and it just hides the saffron color. And so the cell wall is just so thin that the violet just washes out, and that's why the gram negative ends up saffron, and then we can tell the differences. And so I'm going to show you another example here. This is gram negative. So the gram stained purple and human cheeks are cells. So you see gram negative in here and gram positive. So the dark purple, see the whole cell is colored, and then the gram positive is the background of the cell, which is the pink. The gram negative is the background of the cell, which is pink, and the gram positive is the purple from the violet. So you'll definitely see this in my lab. Now I gotta wash this guy off and take steps 2, 3, and 4 coming up next.